Why is it that some features are not visible while using a light microscope? So hello and welcome again, Microbehunter here and Oliver that is, and I'm going to talk about why it's not possible to see some of the features of a specimen using a light microscope, and there are actually three reasons that I would like to talk about. And now I'm not going to talk about the improper use of a microscope, like it's out of focus or the specimen is not centered and so on. So that's not really what I'm talking about here, but rather I'm talking about the physical limits that are imposed by a microscope or by light as such, and basically the physical aspects here. And there are 3 points that I would like to talk about, and I'm going to go through these points point by point, and I'll give you my take on this. Well, one reason why some features are not visible with a light microscope is because those features are simply too small to be seen using a light microscope. There is a so-called a resolution limit. And this means that depending on the objective that you use and also on the wavelength of the light, there is a limit of what can be resolved. And there are certain details that are simply too small. And if you continue to magnify it, for example, by zooming in more, by using a camera or by using a stronger eyepiece, of course, it's going to become larger, but it's going to also become more blurry, and the features are still not visible. And there is a physical law which was proposed by Ernst Abel back in, I think, the 19th century. This gentleman worked at the Zeiss company, and this is one of the most fundamental laws of microscopy, which basically says that there is a certain limit what a light microscope is able to resolve. And this depends also on the wavelength of the light and also on the so-called numerical aperture, which is a property of the objective. I do not want to go into the math now. But there is a limit, and if you want to still see those details, then you have to use a different type of microscope, for example, electron microscopes. Okay, so for example, viruses cannot be seen using light microscopes simply because they're way too small. Generally, as a rule of thumb, the total magnification of a microscope cannot be higher than around 1,000 times. And everything, any magnification that's more than 1,000 times in a light microscope is something that we call empty magnification, which means that you're going to see it bigger, but it's also going to be significantly more blurry and fuzzy, and you're not going to see more. And that's actually one of the problems that especially beginners have with microscopy. And many of them think that the higher that more that you magnify, or the more that the microscope is able to magnify, the more that they're able to see. But that's actually not the case. Just because you make an object appear larger does not mean that you're able to see more details. So that is the most commonly talked about limit, and that is the resolution limit. But that is only one of 3 points, and there are 2 more points that I would like to talk about. Let's say that you should be able to see a certain structure because it is fairly large, but you still have a problem seeing it. And why could this be? And there are 2 reasons for this. And one next reason could be is generally a low color contrast of the specimen to the background. So many microscopic specimens are transparent. And if you now look at a transparent object in water, because in many cases the specimens are in water or in a mounting medium, then you're not able to see very well because the color contrast is too low. And especially a very small object, even if they are pigmented, then because they're so small, the effect of the pigment, the effect of the color sometimes is not very visible. So this is another problem, and one way to solve this problem is either to stain the objects by using certain chemicals that selectively stain certain parts of a cell. For example, there are certain stains that will stain the DNA of a cell very well, and then you're able to see, for example, the nucleus quite well because it stains. This is where you find the DNA. There are also other stains that stain other parts of the organism, and depending on what you want to make visible, you choose a different stain. Now, the problem with that is that this actually kills off, in many cases, the organism that you want to look at. So especially if you want to look at water organisms and they're moving around, you have to be very careful because many of the stains actually kill the organism because, of course, they chemically react with certain parts of the cell. Now, if you still want to increase the color contrast somehow, then you have to use something that you call optical staining techniques by use of filters. And this kind of leads me over into the next, the third point is, and that has to do with the refractive index. And because sometimes even if objects are not clear, are not stained, and if they're clear, if they have a different refractive index, you can still make them visible by using the so-called optical staining. So there are certain filters, for example, dark field patch stop filters or Reinberg filters, or a very common way for difficult organisms, phase contrast microscopy, which requires not only, which requires also special objectives, microscope objectives. In this case, you can actually make differences in refractive index. You convert this into a brightness difference or into a color difference. Basically, this is called optical staining. And basically, this means that if the refractive index of the organism, however, is the same as the surrounding medium, then even those methods will not work quite well. And this is one of the reasons why you have to really be careful if you use unstained specimens, and if you want to rely on phase contrast microscopy, for example, you have to make really sure that the surrounding mounting medium has an appropriate refractive index. Because in the worst case, the specimen would have a same refractive index and also a very low color contrast. So then it's going to become even more difficult. Yeah, those other, sometimes, for example, bacteria, they basically have a very low color contrast. They have a refractive index which is a little bit different than the surrounding water. And even if you do not use any optical staining, if you use a regular bright field microscope, you're still able to see them. Not quite as well, but you're still able to see them because of the difference of refractive index. You're able to see so-called diffraction patterns. And what you actually will see then in the microscope is you will see basically the bacteria, a diffraction pattern of the bacterium. And this basically allows you to see it, even though it is not stained and it's transparent. And even though you do not have any special optics. So there's sometimes these disturbing diffraction patterns, which are actually artifacts, actually sometimes even help you to see objects that normally you would not be able to see. What you have to do is you have to then close the condenser all the way, and this also makes the diffraction patterns much stronger, and therefore certain structures visible. Yeah, okay. So so that's basically the three reasons, which I'm going to summarize again. So the first one is some structures and features are difficult to see because they have a low resolution. They're way below, the features don't have a low resolution. The microscope's resolution is not enough to make those features visible. That is the correct way of actually saying it. Then they're low color contrast. And then also the same refractive index as the surrounding medium. And if basically you have enough color contrast, then you're able to see it, and then the refractive index can be the same. And if this is not possible, then you must make sure that somehow the specimen has a somewhat different refractive index than the surrounding medium. Sometimes actually it's even more complicated than that, because sometimes if you have larger cells like protozoa and you want to look at certain structures inside the cell, then you have of course no possibility of changing the surrounding you can change the surrounding mounting medium if you want to, but actually the object is inside a cell, and then the certain cell organelles are inside the cell, and then the surrounding medium then is now the cytoplasm of the cell, and of course you cannot influence that. So you see that it's quite fascinating. There are many things that you can tweak around here. And these are basically three reasons. Well, I think, that's it for right now. And I wish you all the best, and see you around next time. Bye-bye.